There are various benefits, barriers, and ethics surrounding the xenotransplantation of pigs' organs to human hosts. Xenotransplantation refers to the transfer of cells, tissues, or organs between species and has the potential to overcome the critical need for organs to treat patients, resulting in a possible resolution for the organ shortage crisis. According to many scientific discoveries, it has been found that the pig is considered to be the most suitable animal for xenotransplantation in humans because of its characteristics such as size, the similarity between its organ systems and human organ systems, and its favourable breeding characteristics. Unlike other non-human primates previously used for xenotransplantation, pigs are not an endangered species. According to the Australian Bureau of Statistics, around 5 to 6 million pigs are slaughtered each year for food in Australia. In the US, this number is even higher at around 120 million pigs that are killed for food annually, meaning there would be an abundance of swine to use without creating harm to their species. A major advantage in using pigs for xenotransplantation is the potential to manipulate the porcine genome to create organs that are compatible with the human body. Different genome engineering strategies include gene deletion and knockout. Pigs' genomes encode proteins that are substantially different to the human genome. These proteins carry out additional modifications that do not occur in humans and can prompt harmful immune responses. It is possible to eliminate certain genes, which can help prevent different antigens from being expressed by pig cells. Gene editing. Molecules can be designed to generate double-stranded DNA breaks at any chosen genomic site. After they are repaired, a frameshift mutation can occur which would inactivate the chosen gene. The use of CRISPR technology was recently able to remove and eliminate porcine endogenous retroviral sequences. Gene insertions. Gene insertions help express human genes in pig cells and organs by introducing heterologous DNA into the pig genome. The DNA is transferred into the pig's genome from an external human source. These genes are referred to as transgenes. There are clear benefits surrounding xenotransplantation of pigs' organs to human hosts. Swine are regarded as an ideal candidate when sourcing organs for xenotransplantation in humans due to characteristics and factors such as pig organs are similar to human organs. An example is the similarity between the pig heart and the human heart, as they have a similar heart rate, stroke volume, medium arterial pressure, cardiac output, and blood flow. The kidney of a pig is also similar to the human kidney in relation to its filtration rate and its maximum concentration ability. Another benefit is that pigs are phylogenetically different and do not possess the capacity for emotional responses and altruistic and selfless behavior. The transplantation of pigs' organs to humans presents itself as a promising solution to the human organ shortage crisis. Xenotransplantation has the potential to provide a vast supply of organs that can be selected and obtained very quickly. By using xenotransplantation, there would be a significant increase in the overall patient survival rates and a decrease in the number of patients on waiting lists. There is a big concern about the potential for xenozoonosis infections to emerge in humans due to the transfer of porcine endogenous retroviruses, which are not pathogenic in pigs but are in humans. For xenotransplantation of thoracic organs, size would play a big part as thoracic organs are confined to a closed cavity compared to abdominal organs which have additional room for small growth. Excessive growth could be detrimental to the human host as it could lead to organs outgrowing their blood supply, causing acute organ failure which leads to cortical necrosis. A barrier to xenotransplantation is rejection. Hyperacute rejection is caused by primate and pig antibodies binding together, which leads to a destruction of the graft within 24 hours. By introducing a transgene, it would be less likely to occur. Acute humoral xenograft rejection is related to the deposition of antibodies, and it affects the graft's infiltration, which eventually destroys the graft altogether. Chronic rejection is an immunological injury that evolves from acute rejection, resulting in major damage to arteries, veins, and ducts in organs. Ethically, pigs are presented as an acceptable choice as an alternative source of organs for humans. The processes would not be harmful, yet there would be concerns regarding the harm-to-benefit ratio. The patients would also have to be monitored for possible infectious diseases for an extended amount of time. There is a public health hazard that has the potential to arise, which is the possible transmission and activation of porcine endogenous retroviruses. Xenotransplantation could also decrease the amount of altruistic human organ donors. The outlined benefits and advantages of xenotransplantation of pigs' organs into humans could greatly impact society and help with issues such as the organ shortage crisis. Xenotransplantation from pigs to humans presents itself with barriers that it can overcome, creating a new opportunity that could save lives.